So again, hello everyone. If you might have missed it, my name is Portia Elms and I'm part of the Bridges program for 2019-2020. I'm part of Dr. Cohn's lab, which focuses on immunology and hematology, which is the focuses on the study of the immune system and blood. And today I'm going to be talking about gene editing for common variable immune deficiency, otherwise known as CVID. So to give some background on this, it can occur in both sexes in any ethnicity, and it usually appears a primary form of immune disorders. It is where the immunoglobulin, or some as you might know, antibodies, are deficient, which then impairs the immune system and makes it harder to fight off infections like viruses and bacteria. This is especially important right now because of COVID-19. These patients are super susceptible to having really bad reactions, and they have a really hard time fighting off any infections. And this disease could do a lot of damage. Some of the signs and symptoms are recurrent severe infections, usually occurring in the ears, sinuses, and lungs. Also has been linked to gastrointestinal problems. It also has disease manifestations that include autoimmunity, where the body's fighting off healthy cells, your own healthy cells, and it has increased malignancy predisposition. So the main form of treatment for CVID as of right now is immunoglobulin replacement therapy. This is where they collect a bunch of healthy antibodies from healthy donors, and then they pull them and filter them and give it to the patient by infusion or shot. This is not a cure, but it will stop the reoccurrence of infections. It's given regularly and lifelong. Really big downside, it's an expensive form of treatment, so it's $15,000 to $30,000 annually, and it is for the rest of their life. So the big question is, how is gene editing related to this disease? And in previous studies, they had found that a protein called TACI, in about 10 to 15% of CVID patients, there is a mutation occurring within this protein. TACI is encoded by TNFRSF13B, or I know it's super long, tumor necrosis factor receptor superfamily member 13B gene. And TACI is found on the surface of B cells. And as some of you may know, B cells play a huge role in the immune system. It promotes B cell maturation. It inhibits proliferation. It also will and it also has shown that CVID patients have been described with both heterozygous and homozygous mutations in the TACI encoding gene. And for some people, I've had a lot of visuals because I know I'm a visual learner. We have a DNA strand here. Can you guys see my cursor? Okay, perfect. We have a DNA strand here that has this little red stripe, which would encode for a gene, which is TNFRSF13B, which then encodes for TACI, which is the little surface receptor on this B cell. So our hypothesis for this study was it's two different aims. It's the insertion of a corrective cDNA into TACI deficient stem cells, but then also suppression of the mutated TACI. So we want to suppress the original TACI expression, but then we want to express the new cDNA insertion that we have added. Do I have any questions so far?
Good.
Okay, perfect. And then, so for the first aim, how do we insert the corrected TACI cDNA? It's by a tool called CRISPR-Cas9. If some of you don't know how this works, it's where it has this short guide RNA that can almost act, I'm going to compare it to your Postmates order. You acts as an address for your Postmates driver to drop off your food. Once the sgRNA targets the sequence, or your house, it then binds to the target sequence within the gene. This then allows Cas9 to bind and cut both strands of the DNA. And once the strands are cut, this allows for insertion or deletion, which is the final product. So it would be getting your food. So kind of down here, I have the location or your target sequence. And then sgRNA was kind of little car that takes Cas9 to the sequence. Cas9 gets to the house or the target sequence and will bind and make a double-stranded cut, then result a deletion or insertion or your food. So this is a more scientific way of showing CRISPR-Cas9. As you can see here is the sgRNA and then the Cas9 binding and making these cuts on both strands, therefore having a double-stranded break, and it will have two outcomes, NHEJ or HDR. NHEJ is non-homologous end joining, where it makes a double-stranded break, but the ends just bind back together where it had occurred, as you can see down here. And then HDR is homologous directed repair, where the template will put in place a donor where the break had occurred. And as you can see here, there's a little addition of a new external DNA added to the double-stranded break. And that's what we're looking into now, because we want to add in this new cDNA where the double-stranded break is occurring. So here's kind of like a little background on the HDR progress is we wanted to target exon 1 or 2 for the site-specific gene insertion within the TACI or within the TNFRSF gene. And we began using this reporter donor that has M-citrine, and it's a green fluorescent tag. It kind of just lets us know when we're testing, and in our results, it has green fluorescence and tells us that the donor is in the cells. And we started with M-citrine rather than just going straight into TACI cDNA, because packaging and creating these models for TACI cDNA is really expensive. And also we wanted to see, well, does this work? Are we going to put in a bunch of time and effort? Will it work in the beginning? We found that it did work. It was successful using M-citrine. So then we use a cell line called Ramos cells, which are human B lineage Burkitt lymphoma. And we went on to using a TACI cDNA. So here's a schematic of like the cDNA insertion. Here's the original TACI gene that has mutations within it. And then here you can see within exon 1, or it could possibly be before exon 2, but this one's showing in exon 1. And then here's our TACI cDNA that we've made. And here's how it will be put into the gene. And as you can see, it's still using the original promoter. This is so the cell kind of thinks it's having its original form of transcription and translation, but now it will be transcribing the new donor rather than the old mutated gene. Okay, do you have any questions so far? I know that's a bit of information. We're all good. Perfect. Okay, so here's the second aim of the project. How are we going to suppress mutated TACI? So we want to suppress the original TACI cells, and so this way we can really test our insertion of the new TACI cDNA we're inserting. We use shRNA, which is an artificial RNA molecule with a tight hairpin that will silence a targeted area of the gene. And this is delivered by the packaged double lentiviral vector. And as you know, viruses go in and infect all of the cells and can then give the cell their DNA or mRNA. And this way we've kind of turned off the virus, and now it's packaged with our DNA that we want with shRNA. And the vector takes the shRNA to the host genome. Then once inside the cell, the shRNA will attach to the mRNA strand and silence the targeted gene, which will suppress expression of the protein. So here again, just kind of to see it visually, this is the DNA that gets transcripted into RNA or mRNA and then translated into protein. We're adding at this level a hairpin at the mRNA, which will then no longer allow for translation of the protein TACI. So we're suppressing TACI expression of the mutated one overall. So we had started with just using like singular little hairpins and found that it was not successful. So then in previous studies they had done using multiple hairpins, and we found that that was successful in suppression. And our exact technique we had was using 9 shRNA tandem hairpins. And there's an MNDU3 promoter, and then again using citrine as a reporter to make sure and notify us that the shRNA is within the cells and is actually, that's the way it is suppressing the TACI expression. Do we have any questions so far? All right. So then, so this is like
I
overall.
have a question.
Oh, okay.
There's a question.
Can I see the chat?
Okay,
Okay, perfect.
Nathan has a question. You want to ask the question, Nathan?
Yeah. So his question was, yes, if you use a lentivirus, does your body produce a reaction to it? So honestly, if you're going to be having anything foreign entering a cell, it's going to produce a reaction, most likely a bad one. But it's not producing a reaction where the body thinks that it is a virus. It is just a form of getting our DNA into the genome. It won't acknowledge it as a virus. It's just gonna, we'll see once the DNA enters that it is not a foreign thing such as like a virus where it thinks that the cells need to go and attack it. That is a good question though. Do we have any other questions? All right, so here's the overall idea. We want the shRNA to suppress the original mutated TACI, but we don't want it to suppress our new insertion because that would be kind of counterintuitive. So we made it in a way that it would be able to bind and suppress the original mRNA of the endogenous TACI or the mutated TACI, and it will not be able to bind and recognize the new insertion, therefore not suppressing it, but only suppressing the original mutated TACI. And as you can see here, just kind of further it. And then here are some experiments and results of how we got the cDNA insertion into the cells using CRISPR-Cas9. A question is, how do you even get the cDNA into the cells? And a huge thing that's used in many labs, but specifically a lot in our study, is electroporation. How electroporation works is it's an electrical field applied to the cells, increases permeability of the cell membrane, which then can allow for things like chemicals, drugs, DNA to enter the cells. So here you see this is before you add any voltage or electrical field. The cell membrane is protecting the cell from allowing anything in. Obviously, it allows for water and things like that back and forth, but it is not allowing these like little strands of DNA in. Then when electric field induces a voltage across the membrane, as you can see, there are some of the DNA that is allowed to go in. And you can see, yes, some of the like positive and negative ions are leaving, but that's just part of it once you permeabilize the membrane. And the big thing about this is cells don't like to be shocked. No one likes to be shocked. So they're kind of unhappy after you do this, and they need a lot of time to recover. So taking care of them after is super important. They're just unhappy about having new DNA in them, and then they're also unhappy that was little holes in their membrane. So how our electroporation protocol kind of works is we have like a cell suspension, as you can see them, and then a centrifuge. We centrifuge them, so we spin them down. We then aspirate all the liquid, and you can just see a little pellet of the cells on the bottom. We then add sgRNA and Cas9, as I talked about earlier with the Postmates, and then you can add a DNA donor as well. And then you can electroporate it. And as I was talking about the electrical field, this is the machine we use. It's a neon Thermo Fisher electroporator. It looks kind of weird. It's kind of like a pen, and we just suck up the cells, stick them in this machine, and it zaps them, I guess pretty much is the way to explain it. And then they're plated into a 96 well plate, and it's like a small little area so the cells feel close together. It's more likely for them to recover, and you kind of just have to watch them and give them new media all the time and see how they're doing, because there's a good possibility that they could die. And then if you're using a AAV6 donor, which is a viral donor as well, it's then added now. We don't electroporate a virus because it will be killed in the electroporator. And then we wash and count the cells at 24 hours. And then a big thing is we want to see, okay, did the insertion get into our cells? So we check it by a thing called flow cytometry on day 7, because usually about day 7, the cells have got a little healthier and have recovered a little bit. So how does flow cytometry work? Well, what we do before we run it through this machine, we will stain all the cells with this tag that will tell us if the cell has TACI expression or not. Using an antibody, we specifically use a like PE-RAT antibody. And then it's kind of hard to explain. I barely know how to use the machine myself, but what happens is it puts the sample and it goes through each cell within this like little liquid sample through a fluidic system, then passes through a laser. And these lasers can detect any change or any difference within the cells. It can detect if cells are alive, if cells are dead. We're specifically looking at if the cells have this tag for TACI expression. And then it will send it to a computer and analyze it, and we'll look at the results. So here is our Ramos cells with the neon electroporation on day 7. Here we used a plasmid cDNA TACI donor. And these results are a little bit hard because they're stacked on top of each other, but I'm going to go through it with you guys. And this is the control. And as you can see, it's easier to see on this one. There's 2 peaks. And the first peak is being dead cells, and second peak is being live cells. And this is the control. So nothing had happened to these cells. And, you know, in every experiment, you need a control to see how the cells are acting normally. Then we have an RNP only, where just the sgRNA, the guide RNA, and the Cas9 were added, but no donor. We want to do this to see, is the sgRNA and Cas9 affecting the cells in some type of way that has nothing to do with the donor. And then we have the RNP plus the donor, or the plasmid here. And here you can see there's 2 peaks again. This one is a little harder to see. This is kind of the first peak, and then it goes into the second. But there's a shift. The cells kind of shift to the right, which means there's an increased expression of TACI. So as you see, TACI expressions going this way. So once it shifts to the right, there's an increase. And if it shifts to the left, there's a decrease. And here within exon 1, the shift is not as significant past this little blue line, but here I've just laid it out for you guys. So the control had 59% TACI expression, and a mean fluorescence intensity was just showing how bright the TACI expressing cells are. The RNP had 47%, which was expected because the cells don't like electroporation. And then the RNP plus donor had an increase of 65%, which was, we're looking for an increase, but unfortunately wasn't a huge significant increase, only being about 6%. But the NHEJ, which means cutting is occurring within the DNA, because sometimes a donor could be within the cell, but that doesn't mean it's actually gone to the DNA, has made a double-stranded break, and it had the insertion. And here we can see there was about 70 or 67% of cutting, which is really good. Now within exon 2, the control again was 59%. The RNP was 70%, and we still are not sure. It's still kind of unknown why there was an increase. It could be due to the cells just being stressed overall and then increasing their TACI expression, but we're still not sure, hence why we need a model that has no TACI expression. As you can see here, the control has 59%. We need a model that has 0% to really test how well our cDNA insertion is working, and it's just really hard to tell without a knockout model of the TACI. So the RNPs increased, but then the RNP plus donor is 76%, which is a huge increase compared to the other one, but it's hard to actually accept it because of the RNP being high as well. So we're not sure if it's just the insertion working. Do we have any questions?
I got a question.
Yeah.
So how do you know that I'm not going to say it's wrong, electroporation or whatever the term is.
The flow cytometry?
Electrocuting the cell.
Electroporation?
Yeah. How do you know that that doesn't permanently change the way that they function?
Because it's, go ahead. Okay, so because we're just permealizing the membrane, it will then bind back together. The membrane usually does open and close due to, letting in water molecules, letting out carbon dioxide, like things are constantly going back and forth, but now we're just forcing it to open at like a bunch of spots, then allowing DNA. So it can recover, but also, as you said, how does it recover? How do you know it's acting normal? If it's not, the cells will die. And unfortunately, after electroporation, many cells do die. That's why the recovery process is so important and so crucial to take care of them, because they all just want to die after that point. It's like someone getting electrocuted, you know, they don't really want to live at that point. Did that answer your question?
Yeah, thank you.
Yeah, of course.
We have a chat question too.
Okay. It's harder for me to go to the chat. Here, let me see.
Do you want me to read it to you?
I got it now. Does electroporation increase risk of infection, or does it change their shape and function at all? Would you be able to see if it was possible? So I have looked at the cells right after electroporation, and some of them do usually look different, but they relatively look the same. Cells, when they're getting ready to die, cells are pretty round, and then when they're getting ready to die, they kind of have this like weird shape. So they do sort of look different if they are going to die. And then this is all done in a lab. It's very controlled. The environment's very controlled. So the risk of infection is very low. If our cells were to be infected, it would most likely infect all the cells within the lab. So this is not exactly like a body, you know, like having cells within a body. It's in a petri dish, so it's very, very controlled. And that's why having stuff in research is hard to really correlate to real life as well. I hope that answered the question. And then the next one is TACI present in every human being or only people with the disease? So it is present in every human being. The thing about people with this disease is they have mutations within it. They already have, the thing about this disease is very complicated, but we're not exactly sure all of the genetic mutations that are connected to this disease, but we do know TACI is somehow connected. And it is playing a role within the immune system, hence why they have an impaired immune system. Most of us are pretty healthy. We can fight off a cold. We can fight off infections. So therefore, our TACI is working normally. And then it says Cas9, is there any off-target effect? Of course, you guys have really, really good questions. This is good questions. Cas9, yeah, of course. When making a double-stranded break, there is a chance that it will bind to another part and have off-target effect. We haven't noticed so far any of our stuff having any high off-target effects. Therefore, we're just moving forward. It's been pretty central to the exon 1 or exon 2. Do we have any more questions? I hope that answered your guys' questions.
I have a question if you don't mind me asking.
Uh-huh.
So what prevents the cDNA from being degraded by like the cell's nucleus? Is that like the Cas9 complex that protects it, or how do you avoid the nucleus degradation?
So it can happen. I mean, that's why we're not seeing, if everything were to go perfect, we'd have a huge increase, probably a double increase of TACI expression if all of our cDNAs were completely working, the cells didn't affect them at all, and enhanced all of our cDNA. But as you said, the cell's constantly recognizing that, oh, this isn't mine, and we'll degrade it. But we're just doing it, trying to get it at the chance where we're making this double-stranded break in the DNA and then inserting it, hoping it doesn't notice and it will just bind all together. It's all chance. That's kind of how science works. But yes, the cell does kind of fight against you. It doesn't want to have things that are not originally within the cell, and we're just kind of making this double-stranded cut and hoping that the donor will go in place, but it doesn't always work.
Most of the time you have mixed, like 30% of your cells will work and the other 70% won't. And you can also use the flow cytometry to sort those cells so you can select for the ones that worked and then start with that culture again.
Right. This is kind of why we're doing flow cytometry, because we want to see, did it work and how much of it did it work? And as I had talked about, here, I'll go through it. But here, like, as you can see, this one wasn't successful in percentage of TACI accretion, but there increased, but there was NHEJ, so there was cutting occurring. So double-stranded breaks were being made, but that doesn't mean the donor was going in and working. And we're not sure what that could just be, because it doesn't like to be inserted at exon 1. But here at exon 2, there was a huge increase of TACI expression, but low cutting. So there wasn't as much double-stranded break occurring, but way more of the donor wanted to come in at this place. So it's a lot of hit or miss, and it is trial and error. A lot of the times it doesn't work. I hope that answered your question. All right. And then I have a few more questions on here. It says, is the process of suppressing or making sure that making sure the other type of TACI gene expressed difficult? Does it take a long time to achieve? Yeah, so I kind of go into that later, but we haven't actually tested working both of these models together. This is a kind of new novel thing. People haven't really done shRNA and CRISPR-Cas9 together. We're hoping it will be able to suppress the old mutated TACI and now have a expression of the new insertion. As of right now, we're just testing them separately and seeing do they work at all. But it does take a long time to achieve. I was doing stuff in the lab, unfortunately got cut short because of coronavirus, but I was doing stuff in the lab and I was in there for 7 months, and this is kind of the data I've had so far. So everything does take a long time. It's a lot of trial and error. This is kind of my shining, these are our golden stars, the things that have done well. I haven't showed you guys all the things that didn't work, but yeah, it takes a while. And then how do you select for trans following electroporation? This might be due to how Nicole was talking about we can pick cells. I might need a little more clarification on this question, but pretty much you can pick cells after they've been electroporated using flow cytometry, and then you can go in and it's called a cell sorting machine, and it knows exactly what cells have what, and you can then pick out one or a few colonies of cells and then just grow those cells up by themselves. So then we could always just go in, say, okay, these are showing that these have the insertion, we'll pick these out and then grow them up on their own, but we're trying to completely just change all the cells by electroporation, or seeing how many the percentage of increase we can do with the cDNA insertion. I don't know if that answered your question, but I hope it did.
Yeah, I think it's pretty good. Okay, why don't we move forward, because I'm looking at the clock.
Yes,
so
sorry, just answering. Okay, so experiments and results of the suppression of mutated TACI using shRNA. So how do we get shRNA into the cells? We do a thing collection where we introduce this nucleic acid and all these things together. It's then packaged into a lentiviral vector. And then it kind of acts like a virus. It will then bud out and bud doing transduction into a new cell, and it has foreign DNA introduced by a lentiviral vector. So this will kind of be the host genome. It then lets out all of its mRNA, and this will have the hairpins in it, and then it will bind to the genome, host genome's RNA, and this is where we stop we suppress the mutated TACI protein. So here's our flow results of the rainbow cells transduced with the lentiviral vector of our 9 hairpins. So the first one are untransduced cells. The red indicating untransduced live cells. So those were cells that had nothing done to them. They're just all the live cells. Then these are cells that were stained, as I had talked about earlier, stained with an antibody to show TACI expression. These are kind of showing dead cells and then live cells. There's 67% of TACI expression within the control. This is a negative control. We're expecting no difference really. We know that it's not supposed to have any suppression. So 67 and 65, not really much of a difference. Here's our R9 tandem at an MOI of 10, and the TACI expression took a huge hit. It went from 67% to 30%, and in another replicate 20%. And as you can see, these cells have a huge shift to the left, not really expressing that many of the TACI cells any longer. And then green's showing citrine positive cells. So as we had done, we had a citrine reporter in the shRNA model showing that, okay, the citrine is expressing within the cell. So the hairpins are within the cell. So here's just to hone it in, the overall idea. We're using shRNA to suppress the original mutated TACI, and we did this using transduction. And we use CRISPR-Cas9 and electroporation to insert the new cDNA of TACI. And here's the overall idea of the whole project. Once we're done, this is what we want to do, but we haven't got here exactly yet, is we have CD34 stem cells, which then will have the shRNA, and that will silence the mutated TACI, and that will lead to injection of mice, so the TACI is silenced within the mice. And then we have a CRISPR cDNA insertion, and it will now express the new TACI insertion, therefore having a gene-edited expressed added TACI and none of the mutated TACI. All right, so our future directions is optimizing shRNA to construct for a higher suppression. So it is lower, but we want to have it even lower if we can, maybe 0 if that's possible. We want to create a TACI deficient model using 9-tandem shRNA construct. We also want to combine, as someone had asked earlier, we want to combine both of these tools and see how well do they actually work together. Can it be done? Can they work simultaneously? We're hoping that it can. And then I'd just like to thank Dr. Cohn for allowing me to be in the lab, the Cohn Lab, for all of my questions. A huge thank you to my mentor, Katie Bradford. She's driving force in this, and Cindy Malone for allowing me to be in the Bridges program, my Bridges cohort. Here's my cohort, and here's my lab, and then of course my funding. Do we have any questions? Last questions?